So the beauty of the pyruvate dehydrogenase complex stems from its mechanistic action involving three enzymes and five different cofactors. Namely, the three enzymes are the pyruvate dehydrogenase, the dihydrolipoyl transacetylase, and the dihydrolipoyl dehydrogenase. And then there are various cofactors that play along with those three enzymes, and some of them serve as beautiful arms that facilitate substrate channeling and facilitate the movement of substrates from one enzyme to another in the entirety of the complex. And so if we look at the pyruvate dehydrogenase complex and analyze the three different enzymes involved in this complex, we'll notice that the first enzyme that plays the initial catalytic role with pyruvate is the pyruvate dehydrogenase, whereby pyruvate comes in and a CO2 group is cleaved off utilizing the TPP cofactor thiamine pyrophosphate. So pyruvate comes in and thiamine pyrophosphate has a carboanion that will be utilized in a nucleophilic reaction with the pyruvate molecule. That is, there will be a nucleophilic reaction such that the electrophile here is the pyruvate's carbonyl group, and then a CO2 group will be cleaved off in the process. So this forms a hydroxyethyl TPP, and the hydroxyethyl TPP will then be utilized in the next reaction as a reactant, whereby the lipoamide arm of the second enzyme, dihydrolipoyl transacetylase, will that lipoamide arm will swing forth to the enzyme 1 component of the pyruvate dehydrogenase complex, and it will be such that the acetyl unit or the 2-carbon unit of the hydroxyethyl TPP will be transferred onto the lipoamide arm, and this facilitates the next step, which is the lipoamide arms swinging back to the enzyme 2 component from the enzyme 1 component and transferring that 2-carbon unit onto the coenzyme A. And then this transfer onto the coenzyme A will facilitate the production of acetyl CoA, and thus we have a product that is now ready and generated and can be released. And then acetyl CoA has various fates. It can enter the citric acid cycle. It can be utilized in fatty acid synthesis. It can be utilized in ketone body formation. It can be utilized in cholesterol synthesis, and so forth and so on. But generally, the main view whereby this is discussed is in the view of energy production. And so the next main step is the citric acid cycle for that acetyl CoA, whereby the acetyl CoA is combined with a 4-carbon oxaloacetate to produce citrate via citrate synthase. But back to the pyruvate dehydrogenase complex, what's ought to be recognized here is that we have now produced a dihydrolipoamide arm that needs to be reoxidized. And this reoxidation is something that is handled by the dihydrolipoyl dehydrogenase component or enzyme 3 component of the pyruvate dehydrogenase complex, whereby the dihydrolipoamide arm is going to be oxidized through the reduction of an FAD molecule, and this FAD molecule will then reduce an NAD plus molecule, producing NADH. And what's ought to be recognized here is that usually this happens the other way around. That is, an NADH molecule can reduce an FAD molecule due to the reduction potentials and the nature of their oxidation reduction abilities. But in this case, we have an FAD molecule that is being reduced first. That is the FAD cofactor that is reduced, reoxidizing the lipoyl arm and facilitating the ability of that lipoyl arm to swing back to the enzyme 1 component and engage with another hydroxyethyl TPP that's been formed by another pyruvate molecule. Now, what's ought to be recognized here is that once you have the NADH produced, that NADH molecule serves as an energy currency molecule, enter oxidative phosphorylation as well. But now let's discuss the citric acid cycle and delve into that. Now, what's ought to be recognized here is that once we have the acetyl CoA can be shuttled into what's called the citric acid cycle, or the TCA cycle, whereby the very first step is a condensation reaction whereby citrate is produced through the condensation of oxaloacetate with the acetyl CoA. And then you have a CoA leaving group that's going to leave. That is, the coenzyme A attached to the acetyl group will leave in this condensation reaction and will produce citrate. Now, once we produce citrate, we'll isomerize citrate into isocitrate via aconitase. And then once we have isocitrate, we're going to have the very first oxidative decarboxylation. So isocitrate is going to be oxidized and decarboxylated to form alpha ketoglutarate. This produces NADH and CO2 in the process, and this is catalyzed by isocitrate dehydrogenase. So this is a pivotal step because it's the first oxidative decarboxylation that happens in the TCA cycle. And then immediately after that, we're going to have the second oxidative decarboxylation, whereby the product that is produced, alpha ketoglutarate, will undergo an oxidative decarboxylation. This will be the second oxidative decarboxylation, whereby we're going to form succinyl CoA, and also we're going to produce NADH and CO2 in the process. Obviously, this is a decarboxylation reaction. It's also an oxidative decarboxylation reaction, so we're oxidizing the reactant at hand, which is alpha ketoglutarate, and reducing something else in the process. As we know, oxidation and reduction reactions are always coupled. And so what we're reducing in those two oxidative decarboxylations are NAD plus molecules and producing NADH in the process. And those NADH molecules can go on to the electron transport chain so that they can engage in oxidative phosphorylation. Now, a key step that's going to happen next upon the generation of succinyl CoA is a substrate level phosphorylation. That is, succinyl CoA is converted into succinate, producing a GTP or an ATP molecule. They are somewhat equivalent with the help of succinyl CoA synthetase. And so succinyl CoA synthetase will be the enzyme that catalyzes this reaction, producing a GTP molecule or an ATP molecule and producing succinate in the process. Now, once we have succinate, the next step is the dehydrogenation of succinate. So succinate is oxidized to fumarate, and this generates an FADH2 molecule. This is an oxidation reaction, ought to be coupled with a reduction reaction. And the reduction reaction that is happening, or the reduction that is happening, is that of the reduction of FAD into FADH2, and this is catalyzed by succinate dehydrogenase. Once we have fumarate, we're going to hydrate fumarate into malate. This is catalyzed by fumarase. And then once we have malate, the next step is another dehydrogenation reaction that produces NADH, an NADH molecule. And this is catalyzed by malate dehydrogenase, and it's going to produce an oxaloacetate. And thus we have regenerated an oxaloacetate, and we have produced a cycle. That's why it's called the Krebs cycle or the TCA cycle, because it is a cycle whereby we regenerate oxaloacetate that's now ready to interact with another acetyl CoA, produce a citrate in the process via citrate synthase, and so forth and so on, in a cycle that produces all these high energy electron carriers that can go on to the electron transport chain. And let's not forget about the substrate level phosphorylation, whereby we produce a GTP or an ATP molecule in the process. Before I delve into the regulation of the TCA cycle, let's recap how many molecules we produce from one acetyl CoA. So we produce 2 molecules of CO2. So we have completely generated CO2 molecules from the 2 carbon units of the acetyl CoA. And we've also produced 3 NADH molecules and 1 FADH2 molecule. And then we produce a GTP or an AT molecule as equivalent. So let me talk about the regulation of the TCA cycle. And so the regulation of the TCA cycle happens through various different mechanisms. And the first mechanism that's very intuitive is the relative levels of ATP to ADP. So the relative levels of adenosine triphosphate to adenosine diphosphates. And so in the cell, if you have high ATP levels, and this will indicate sufficient energy, slowing the TCA cycle. Well, if you have high ADP levels, and this will indicate need of energy, stimulating the cycle. Now, this happens via various regulatory enzymes in the TCA cycle that will respond to ATP ADP ratios. There's also the NAD plus and NADH ratio. So if you have high NAD plus, then this indicates that you are needing of energy-rich molecules, in need of energy-rich molecules, and this will stimulate the TCA cycles. On the other hand, if you have high NADH levels, then this will inhibit the cycle. There's also allosteric regulation. So key enzymes such as citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase are regulated allosterically. For example, citrate synthase is inhibited by ATP and NADH and activated by ADP. So citrate synthase, if you recall, is the very first enzyme responsible in the TCA cycle for that condensation reaction that's going to involve oxaloacetate and acetyl-CoA. And so what's ought to be recognized here is that this key enzyme is also a key regulatory enzyme that will respond to ATP and NADH levels. It is inhibited by ATP and NADH molecules, and it is activated by ADP molecules. Furthermore, the cycle intermediates such as citrate and succinyl-CoA can inhibit enzymes earlier in the cycle. This prevents the overproduction of those intermediates. And also, calcium ions play a role. So, for example, in muscle cells, calcium ions act as a signal for increased energy demand and activate isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase, enhancing the cycle's activity. So, it's also to be recognized here are the three key enzymes in the TCA cycle that are involved in the regulation of the cycle. So, these include citrate synthase, as I just mentioned. This is the very first enzyme in the TCA cycle, and it catalyzes the very first step, and it is regulated by feedback inhibition from ATP and NADH, and also one other molecule, and that is citrate itself. So citrate itself can inhibit citrate synthase, and so this is feedback inhibition by ATP, NADH, and citrate of the citrate synthase enzyme. Another enzyme that's a key regulatory enzyme in the TCA cycle is the isocitrate dehydrogenase. This controls the rate of the cycle through allosteric activation by ADP and inhibition by ATP and NADH. And then you have alpha ketoglutarate dehydrogenase. So if you recall, isocitrate dehydrogenase is involved in the first oxidative decarboxylation, step of the cycle. Then you have alpha ketoglutarate dehydrogenase. This is another enzyme that's involved in the second oxidative decarboxylation step, and it is inhibited by its products, NADH and succinyl-CoA, and activated by calcium ions in muscle cells. And so what ought to be recognized here is that you have three key regulatory enzymes in the TCA cycle. These include citrate synthase, the very first enzyme, and also the two enzymes involved in oxidative carboxylations. Now, what's ought to be recognized here is that the intermediates of the TCA cycle can undergo various different fates. That is, these fates of the intermediates of the TCA cycle can be anabolic in nature or catabolic in nature. And so citrate, for example, can be exported to the cytosol where it's converted to acetyl-CoA for fatty acid synthesis and cholesterol synthesis. Alpha ketoglutarate serves as a precursor for amino acids like glutamate, which can be further converted to other amino acids or used in neurotransmitter synthesis. Succinyl-CoA is involved in heme synthesis, and it's an essential component of hemoglobin and other hemoproteins. Malate can be converted into oxaloacetate in the cytosol, which serves as a precursor for gluconeogenesis, production of glucose from non-carbohydrate sources. And oxaloacetate can also be utilized in various ways. So it can be utilized in gluconeogenesis or converted to malate and then pyruvate, providing substrates for additional energy production. Now, we understand that the electron-rich or the electron energy carriers, NADH and FADH2, will go on to the electron transport chain and donate their electrons. These electrons will be donated in the inner mitochondrial membrane. Specifically, NADH transfers electrons to complex 1, while FADH2 will donate its electrons to complex 2. Now, electrons move through a chain of protein complexes, that is complex 1 through 4, and mobile carriers like ubiquinone and cytochrome C. And what happens is energy is released as those electrons move through the chain of protein complexes. And this energy is utilized to produce a proton gradient. This proton gradient is then further used by ATP synthase to convert ADP ADP into ATP. And so proton gradient, which forms the proton motive force, as it is called, drives protons back into the matrix through the ATP synthase. And this flow of protons provide the energy for ATP synthase to convert ADP and inorganic phosphates into ATP. And what you have to recognize here is the final electron acceptor is oxygen. And this final electron acceptor, which is oxygen, when it accepts electrons, will and combines with protons, will form water. And this is a crucial step for maintaining the flow of electrons through the ATC. So you have to have a final electron acceptor such that the electron transport chain is not backclogged, and so that it can continue to run as more energy or electron carriers come into the electron transport chain and donate their electrons.